Hello everyone, this is Joyla Maricazos, and for today's video, we're going to do an experiment which is to extract and examine my own DNA in my cheek cells. But before we start any experiment, we have to make sure we have all our materials to extract our DNA. So for this experiment, first we will need table salt, also known as sodium chloride, warm water for the saline solution for rinsing my mouth, a disposable cup where we can put the salt water, and another disposable cup to put our gargle solution, some regular dishwashing soap, pineapple juice which will act as an enzyme, a dropper, stirring rod to mix our solution, and a cold rubbing alcohol, also known as 70% isopropanol. So for the next part, we will go over with the experiment. So let's start. So this is how the process works. So first, we will need to prepare the saline solution. So we will add 1 tablespoon of sodium chloride, also known as table salt, with 2 cups of hot water. So it is important to stir the solution continuously to get the salt completely dissolved.
And now we're done stirring it, and I think there is not much undissolved salt left. So the solution is now clear and ready.
So now we will have to gargle 1/4 cup of this and gargle it for a whole minute.
So now we have here 1/4 cup of my cheek cells mixed with the saline solution, and now we will add 2 drops of dishwashing soap. Then we will stir it gently to make sure we do not create bubbles in the solution. Next, we will add a pinch of pineapple juice to act as an enzyme that will cut the proteins and will separate the DNA from the protein. After we have stirred in the pineapple juice, which causes DNA to separate, we will then need to add cold 70% isopropanol or rubbing alcohol. So as we all know, DNA is soluble in water but isn't soluble in isopropanol. Since isopropanol is miscible in water, then what we're essentially doing is lowering the DNA solubility of the solution. And when it has sufficiently lowered, DNA will precipitate. So now let us wait 3 to 4 minutes to see if our experiment successfully works and we can see any DNA. So it has been 4 minutes now, and if we check the solution, we can see that it formed a fibrous DNA right there. It is not much, but some actually precipitates. And if we look at it in this angle, we can see that the DNA floats and there is a separation between saltwater solution and the alcohol. And we're done! We successfully extracted some of my DNA, and I hope that you learned something from this experiment. Again, this is Johaila. See you in my next video.